Cancer Cell Biology Part 2. In Part 2, we are going to look at some examples of proto-oncogenes and oncogenes. And in Part 3, we are going to look at some example of tumor suppressor genes. So in the first part, we talked about oncogenes and proto-oncogenes. Just as a reminder, when a proto-oncogene becomes mutated to an oncogene, it can contribute to neoplastic phenotype. These are always gain-of-function mutations, which means the product of the oncogene is much better at driving the cell cycle than the product of the unmutated gene. And these mutations are often referred to as dominant because you only need a mutation in one of the diploid pair of these genes to make a contribution to transforming a normal cell into a tumor cell. There are many different ways in which you can acquire a gain-of-function mutation, and some of the examples that are on this slide. First of all, on the far left, a change in the coding region of the gene may occur. It could be a deletion, it could be a point mutation, it could be an addition. So there is a small change in the coding region such that once this mutated gene is transcribed and translated, you end up with a different protein that is hyperactive, a protein that's better at driving the cell cycle than its normal counterpart. The other way to gain of function is gene amplification. So normally we have 2 copies of every gene, but you can get amplification of genes such that you now have 3 or 4 copies, and when all of those copies are transcribed and translated, you end up with a lot more that protein. So the protein is not mutated, it's normal protein, but there is just a lot more of it driving the cell cycle. And of course, that's also a gain of function. In cancers, you see a lot of gross chromosomal rearrangements, and where you can get a piece of chromosome breaking off of one chromosome and translocating and attaching itself to other chromosomes. Or you get balanced translocation when you get exchanges between 2 chromosomes. Part of one chromosomes are exchanged for parts of another chromosome, and I will show you an example in a moment. But chromosomal rearrangements can be very detrimental. So for example, when a proto-oncogene is normal chromosome, there is a promoter that is driving its expression. But that promoter is highly regulated. It is kept off except in the specific part of the cell cycle where it is needed. There is a possibility that though, that chromosomal translocation, that proto-oncogene can end up downstream of a promoter that is unregulated, a promoter that is on all the time, a constitutively active promoter. And in that case, you would get a strong overexpression of that protein. So again, in this, the protein would be normal. The protein itself is not mutated, but there is just a lot more of it there. Another possibility is that you can get a part of the proto-oncogene or all that proto-oncogene attached to another gene, and so you can end up with this strange new fusion gene. And when it's transcribed and translated, you end up with a brand new protein, a fusion protein that might be constitutively active or hyperactive. So let's look at a couple of examples of these proto-oncogenes. So the class 2 proto-oncogenes are the growth factor receptors, and we talked about that in part 1. I just want to remind you the most common member of this class are the receptor tyrosine kinases on the cell surface. So let me just remind you how the receptor tyrosine kinase work. You know that they are normally monomeric. They have an extracellular domain that binds ligand, and they have a transmembrane alpha helical region and a cytosolic domain that contributes to the kinase catalytic site. And when you get ligand binding, you get dimerization of that receptor. You get cross-autophosphorylation, and the phosphorylated receptor can then, through a series of steps, activate RAS, which in turn activates RAF, which in turn activates MAP kinase, and then MAP kinase can move into nucleus and phosphorylate transcription factors and drive the expression of genes. And often these genes are positive regulators in cell growth. So the receptor tyrosine kinases are important proto-oncogenes. Here are some examples in colon cancer. In many cancers, there is a receptor tyrosine kinase that is implicated. Here is an image in its normal state. So this is a product of proto-oncogene. So it's not mutated, and this receptor is known as the TRK receptor. And it is a typical tyrosine kinase receptor with an extracellular domain and transmembrane domain, and the kinase domain, and it behaves exactly like the receptor tyrosine kinases. It binds to the ligands, dimerizes, activates RAS, and so on. There is another partner protein that is implicated in many colon cancer, and that is a non-muscle tropomyosin. And its normal states is shown in the right here, on the right-hand side of your slide. This is a dimer that is made up of two helical interaction between two parallel alpha helix monomers. In many colon cancer, there is a chromosomal translocation, and that connects the part of the tropomyosin gene to the TRK gene. And you end up with this very unusual fusion gene inside of these colon cancer cells. And the product of that fusion gene is shown right here on the bottom of your slide. And it's a very unusual protein. It contains a part of this tropomyosin shown here in blue, and it contains a part of the receptor. There is no extracellular domain, and this unusual fusion protein is a chimeric protein, and it's completely cytosolic. It is not trafficked to the plasma membrane like this. But what happens is that you get spontaneous dimerization of these tropomyosins. The dimerization brings the two kinase domains in close proximity to each other, and that activates it, and you get cross-phosphorylation. And then you get downstream signaling, you get the constitutive signaling, and that actually contributes to the phenotype of colon cancer cells. So here is an example of a mutated cell surface receptor, and the mutation is such that it contributes to the transformation of the cell. So in breast cancer, especially the most metastatic breast cancers, there is a receptor known as HER2 receptor that is involved. It is shown here in its normal state. Again, it is a typical receptor tyrosine kinase. I need to point out that in the alpha helical region of the transmembrane part of this protein, there is a key valine amino acid. And in the most aggressive breast cancer, this valine is converted to glutamine. And this mutation leads to a constitutive dimerization of these domains in the absence of the ligand. So there is a change in conformation such that you get autodimerization without the ligand. You get cross-autophosphorylation and the constitutive signaling. And this contributes to the phenotype of breast cancer cells, the cell cycle phenotype of breast cancer cells. In many other cancers, like epidermal growth factor receptor is involved. Again, this is a typical receptor tyrosine kinase. In many cancers, you do see a truncation mutation such that you lose the large portion of the extracellular domain as shown here on the bottom right. And for the same reason, when you lose your extracellular domain in these receptor tyrosine kinase, a conformational change occurs, which results in a constitutive dimerization and autophosphorylation, which results in a constitutive signaling from these receptors in the absence of ligand, and that will lead to the neoplastic phenotype. So let's take a look at the intracellular transducers, which are the class 3 oncogenes. An example I want to look at is RAS, because we already talked about RAS a lot. So let me remind you how RAS works. It is a binary molecule. It's a molecular switch, which means it exists either in the on or off conformation. When it's bound to the GDP, it's off, and when it's bound to the GTP, it is on. And the internal GTPase activity of the RAS, the intrinsic GTPase activity of RAS, can cleave the GTP to GDP, and in doing so, it turns itself off. It's usually needed the help of a protein known as a gap, but gap is not what is hydrolyzing the GTP to GDP. The gap is just actually turning on the GTPase, the intrinsic GTPase activity of the RAS itself. In over 50% of the cancer, there is a mutation in RAS, an amino acid in the position 12. It is a glycine to valine mutation, G2V, and we talked about this as the V12 mutation in part 1. And what happens is when you have a G2V mutation, a G12V mutation, you abolish the intrinsic GTPase activity of the RAS. So if the RAS does not have the GTPase activity, it can never hydrolyze the GTP to GDP, which means it is always on. This is not the only mutation in the RAS that you see in cancers. There is a Q67L mutation, a glutamine to leucine mutation at the 67th position. You abolish the intrinsic GTPase activity of the RAS, so the RAS becomes constitutively bound to GTP and it can never turn itself off, and you can see how very detrimental that is. RAS normally is activated by these receptor tyrosine kinases, but if it is constitutively active, it doesn't matter what is happening upstream, right? The RAS is on all the time. It will always signaling to RAF, which will signal to MEK, to MAP kinase, and it will drive the transcription and turn on the genes that regulate the cell growth. And so it is clear to see that how the cell cycle can be dysregulated by having the RAS that is permanently turned on in a cell. And then let's look at one example of a transcription factor, which is a part of class 4 oncogenes. The cancer that I want to focus on is Burkitt's lymphoma, which is a B-cell tumor, a B-cell neoplastic transformation. In Burkitt's lymphoma, there is a balanced translocation between the chromosome 8 and chromosome 14. And what I mean by this, that a portion of chromosome 8 and a portion of chromosome 14 are exchanged, and they are exactly the same size. That's why it is called the balanced translocation. And so there is no change in the size of these chromosomes. There is just an exchange of one part. But there are some important regions in these exchange parts. On chromosome 8 is a transcription factor that is known as a MYC. We talked about this in part 1 already. And where it sits on the chromosome 8, its expression is highly regulated. So it's only turned on in the part of the cell cycle when its gene function needed, and then its function is rapidly degraded. So the appearance of MYC itself is very transient. On chromosome 14 are some genes that are constitutively on in B cells that make up the parts of the antibodies. Each antibody is a Y-shaped molecule where the top V-shaped part is made up of two chains, variable heavy and light chains, variable light chains, VL, where the stalk is made up of a constant heavy chain, or CH. Remember, the B cells are responsible for producing the antibodies. You should remember this if you have taken immunology or microbiology. So these are the parts of the genes that when you have transcribed and translated, they become antibodies or chains of antibodies. And as you can imagine in B cells, since it's job of B cells to make antibodies, these are on all the time. So they are sitting behind very strong promoters. But the MYC is sitting behind a very regulated promoter, which is only turned on very briefly in the cell cycle. So these genes, these immunoglobulin genes, are sitting behind a very strong promoter that are on all the time. When you get a balanced translocation, you actually end up with the MYC sitting right behind the immunoglobulin gene enhancer and the promoters. And that permanently turns on the MYC. And MYC is now a positive regulator of the cell cycle. So you end up with a lot of MYC in the cell, which rapidly and continuously drives the cell cycle and contributes to the formation of the tumorous phenotype. So this is also a gain of function, and this is the class 4 transcription factors. So this completes the part 2 of cancer cell biology.